Now let's finish up our mitosis-meiosis lab by actually looking at some cells undergoing mitosis. We can't just grab any clump of cells and expect to find cells that are dividing. We need to look in some special places. In plants, they grow at the tips, the tip of the stem, the tip of the root, etc. And so we're going to use onion root tips. Excuse me. Just below the root cap, there's a little region we call a meristemic region. A meristemic region means there are cells that are still capable of dividing. And so typically in lab, we would have some onion root tips for you that had been harvested at around noon when there's a peak in mitosis, add a chemical that stops the spindle fibers. And so the cell is stalled. Whatever stage it was in when we added this chemical called colchicine, that's where the cell still is. So when we make a slide of the onion root tip, we hope to see cells in interphase, but also a lot of cells in the different stages of mitosis. Here's what a typical cell would look like, and here you can see some of the stages of mitosis, anaphase, metaphase, etc. There's a telophase, a couple other metaphases. There's some more. Let's take a look at this one. This is a very nice picture of an onion root tip showing you the different stages of mitosis. So, which of these cells, A, B, C, or D, do you think is in prophase? I hope you said D. All right, what about metaphase? Metaphase is actually A. I know it doesn't look like a perfectly straight line, but remember that metaphase is happening in 3 dimensions. And so the cell labeled A is in metaphase of mitosis. Anaphase is pretty obvious. I hope you can see that the cell labeled B and the one below it are clearly in anaphase. The sister chromatids are being pulled apart. And lastly, in C, we see telophase. There's a little bit of a cell plate forming, and you'll see a better picture of that in just a little bit. Now, on page 105 in your lab book, they ask you to count 50 cells from a root tip slide and count how many are in interphase, how many are in prophase, metaphase, anaphase, and telophase. Interphase is easy to spot because you can often see a nucleolus in the nucleus. So if you see a dot on the nucleus or 2, that means the cell is in interphase. Prophase, you'll just start to see the cells condense. And so you'll just start to begin to make out those chromosomes. And then, of course, in metaphase, they're lined up, etc. Now, if you look here, I can see a couple cells in telophase, I see an anaphase, I see all sorts of things. So go ahead and count up 50 of the cells, how many of each of the different stages. Now, animals don't grow at the tips. We grow sort of from the inside out. And I can't just grab a clump of cells from an animal and expect to find cells undergoing mitosis. There is one time when your cells are dividing more rapidly than they ever will again, and that is in early embryonic development. This is a picture of a whitefish embryo, what's called the blastula, and it shows you a couple of different stages of mitosis. Often within the blastula, the cells are pretty much synchronized, and they're all dividing pretty much, they're in the same stages, I guess is what I'm trying to say. So here's a nice picture of the different stages of animal mitosis. You can see prophase, you just start to see those chromosomes. Metaphase, lined up down the middle. Anaphase, starting to be pulled apart. Telophase, the chromosomes start to uncondense, and so they start to get long and thin again. And then later on, we're going to start to see cytokinesis. Cytokinesis is the division of the cytoplasm that occurs after telophase, if we're being strictly correct. Most people just clump it in with telophase, frankly. It's different in plants and animals. It has to be different in plants because they have a cell wall. And so a small cell plate forms. Eventually, that will indeed become a cell wall. Animals, we just have a pinching inward of the microtubules, and you can see there what we call a cleavage furrow. So cytokinesis, different in plants and animals. Now, your professor might want you to know some of the differences between spermatogenesis and oogenesis. Spermatogenesis begins at puberty and goes for life. And millions and millions and millions and millions of sperm are made every single day. For each what we call primary spermatocyte that forms, 4 sperm are going to result. Sperm are very small cells, and males make lots and lots of sperm every single day. Oogenesis, egg production, is very different. For each primary oocyte, we're actually only going to get one egg cell or one ovum. Cytokinesis is very uneven, and one cell gets almost all the cytoplasm. Can you think why? Just take a minute and think why eggs are so big. Well, you get all of your cytoplasmic machinery in the egg. And also remember, other than mammals, all of the energy, all of the food for pre-hatching development needs to be there. So egg cells are very large and filled with nutrients. Only mammals rely on the female to take care of them. Okay, there you have it. There it is. Now, what you should be doing, I would suggest at this point, is starting to work your way through the lab and work on your questions. You want to begin on the top of page 102 by drawing that cell in gap 2, and that should be easy for you, I hope. Work your way through the stages of mitosis, meiosis with the chromosomes that you made. I hope you did make some chromosomes from Play-Doh or from pipe cleaners / chenille stems, or even paper, something. It really helps if you physically can move the chromosomes. Watching a PowerPoint is not the same as actually moving the chromosomes yourself. I also think you'll find that 2-page handout, and I have posted it. It's the first thing posted in this module. I suggest that you have that open or even print out a color copy if you can. My students find that figure very, very helpful while they're answering the questions. They can look at the cell in metaphase of mitosis. They can compare it to a cell in metaphase 1 of meiosis. Just a really nice figure, and it'll really help you out, I think. Well, I enjoyed doing the mitosis meiosis lab with you. See you soon. Bye-bye.